Our next speaker is Dr. Conchita Alonso, who's coming to us from Spain, where she works at the Estacion Biologica de Doñana near Sevilla. And she works on ecology and evolution of plant-animal interactions, on ecological epigenetics and pollination. And the title of her talk today is A Dynamic Epigenetic Perspective on Above and Below Ground Phenotypic Responses to Drought.
Okay, thank you very much for the introduction, and thank you, the organizer, for launching this initiative and giving me the opportunity to present here some of the studies conducted in our research group that pay attention to the significance of...
So we cannot hear you very well.
Conchita, your voice is very quiet. Can you speak a little louder?
Sorry. Is it now better?
A little bit, but it's still kind of quiet.
Okay.
Is it now better?
Yes, that's better. Thank you.
Okay, perfect. Then we continue. So yeah, this is the award that I have been doing with Mónica Medrano and Carlos Herrera, and the help of some of our lab technicians and several people, students that help for the practical greenhouse experiments. And the outline of the talk is this. I will make a brief introduction about epigenetics, then I will show you some of the results of an experimental study, and I will wrap up with some take-home message and future perspectives. Well, with this graph, I would like to introduce you why I became interested in epigenetics and why I think that plant ecologists should pay attention to this. As ecologists, we are interested in phenotypic variation, and we can understand phenotype as the interaction between genome, here represented in orange, and the environment. And in this view, epigenetic variation appears like an extra intermediate layer between the genome and the phenotype that for sure contributes to increased phenotypic variation. And I would like to highlight three features that distinguish epigenetic variation or variability from genetic variation or genetic mutation. The first one is that at least some of the epigenetic variants can be triggered by the environment, and this means that many individuals in a certain place could develop the same variants, and that's a huge difference for natural selection to occur. They are also transmissible from parents to offspring, but they don't last forever, so they can be reversible. And this is also a huge difference with mutations, because they provide somehow a memory of past interactions between the genome and the environment, but still they are much more dynamic. And they are also very much variable, and this has been proved in model plant species when people have estimated the epimutation rate of the changes, and is several orders of magnitude higher than mutation rates. And also we have found that in natural plant populations, when usually we estimate with anonymous markers, for instance, diversity of epigenetic variants is much higher than genetic variability. There are several mechanisms that contribute to epigenetic variability. All of them, DNA methylation, histomodification, and small RNAs. Nowadays, we know that they are interrelated to each other, and I will be talking to you just about DNA methylation. That is one of the best known in plants and is highly variable across species, across populations, across individuals, and even within plants. So I feel that this is one of the most important for plants. Well, this is my study area, mountainous area in southeastern of Spain in a Mediterranean climate. The weather there is hot in summer and dry, and it can snow in winter. So we have a really unpredictable and stressful climate both in summer and winter time. It's heterogeneous in the landscape, and is a place in which there is a rich community of endemic plants. And in the graph here, you can see, for instance, that the precipitation is seasonal and highly variable across years. So we think that the plant community there is in risk because we have seen today a lot of studies talking about increase in temperature, and we already have passed the 1 degree increase relative to the all ages. But in all the predicted models, when the temperature increase, the time under severe and extreme drought is also going to increase. And these are graphs that for the expected time that our places will be in severe extreme drought, even if we just grow up or increase temperature by 1.5 or 2 degrees, we are facing this. So my study will pay attention in particular to drought, and we will use a drug, a chemical compound that is 5-azacitidine that it has been proved to modify the methylation of the DNA in several organisms, including plants. And well, I think that this is the scheme of the study. So the questions that I would like to address is whether changes in DNA methylation will be associated to plant phenotypic traits that are relevant for fitness, those changes that we induce with the chemical, also if drought will modify DNA methylation, and also phenotypic plant traits that are relevant for fitness, and if epigenetic is relevant for the response. We expect that the individuals that have been raised their system will respond differently, and then we will find some interactions. So this is the model system that we have searched. Is Aerodium secutarium. It's an annual herb that is flowering in the spring. It's native to our region, but it has become also invasive in many places worldwide. And we collect seeds from 2 different plant populations, one that is flat and has a deep soil, and another Puerto del Tejo that is more dry and has a more rocky soil. This is the experimental design. So as I said, we collect seeds from several plants in the field, then we grow a first generation in greenhouse to homogenize the past experience of the individuals, and then I will tell you about a second generation experiment in which we have 2 factorial factors to the methylation treatment and the water stress treatment. And I measure as a genetic or epigenetic, I measure global DNA cytosine methylation, both in leaves and roots, at seedling stage and adult stage, and several phenotypic traits, and they have been analyzed with linear mixed models, including family as a random factor. So the first step is to prove that the treatment that we are doing with the seeds, here you can see a graph or a picture where seeds were embedded in this chemical compound, was effective. And we found that at seedling stage, DNA, here in this graph, the global methylation level is in the y-axis, and you see that the DNA was more methylated in leaves that are in blue than in roots that are in yellow, and that the treatment was more effective in the leaves. However, when we measure this in the adult stage, and here you see the picture of the final stage of the plants, and when we sample the roots, and this is and a leaf that was collected at flowering time, then we see that at adult stage, DNA was more methylated in roots than leaves, and the effect of azacitidine was much more evident in roots in the yellow graphs. So this means that epigenetics and DNA methylation is highly dynamic and changes with age of the plant, and also is variable between different tissues. And this is something that usually has not been addressed. Most of the studies that have been conducted mainly analyze leaves. So the effects of the impact of the treatment in the phenotype, you can see here, it control plants in green and those treated with azacitidine in yellow. And we can see that there is a delay in flowering. And well, there is also a significant effect of the population of the source population. And but when we apply the drought, what we see is that drought also delay flowering, but it only delays flowering in the plants that flower later and that have a standard or a natural level of methylation. So there is some interaction between the two treatments in this particular phenology trait. If we now look at the fitness as, for instance, the total number of inflorescences produced, that as it is an annual plant is a good proxy, we found that the methylation and drought both reduce the total number of inflorescences produced, and we also found that the plants in CH population produce less inflorescence. And another interesting trait is the final biomass, and the most significant result was with the root biomass, also with above ground. But in the graph I showed you root biomass, and we see that drought reduce the biomass. This is not really surprising, and but azacitidine has no effect in that. And again, we have an effect of the population of collection. So it was really important to see if the drought had an impact in the DNA methylation. And we found that yes, in leaves the effect was mild, but drought erased the effect of demethylation that we have seen in the two populations. But when we look at roots, the effect was much more stronger, and drought reduced methylation, so both treatments have the same direction of effect, and the response was lower in the azacitidine treated plants only in these PT populations, the one that has a stronger decrease. And for us was very interesting to see that what happened was that drought altered the methylation within plants, like a coordinate individual, because when they are in control, we have found a positive correlation and highly significant, so that individuals that have a high level of leaf global cytosine methylation, that is in the x-axis, also have that in the roots, and roots are usually more methylated at adult stage, as I said at the beginning. But when the plants are in drought, in the right panel, the correlation disappears, and this suggests that the regulation is not, is to some extent at least, is independent above and below ground. And if we pay attention to what happened in the two different populations, we can see here now in 4 panels that is in the PT population, in the population in which plants grew smaller and also flower earlier, where the synchronization of the two tissues is stronger, and they manage to produce in the end a higher fitness. So to sum up, we conduct this experiment aiming to understand if epigenetic regulation could help to deal with the recurrent stress. And what we found is that drought induced the methylation in root DNA. So in principle, yes, that the methylation and drought both delay flowering, that in principle will not be really beneficial, and that because late flowering plants produce less inflorescence. And how does it work? It's not homogeneous within a species, but, and this is important because we can think that all populations could respond similarly, and it was not the case, but early flowering plants decouple methylation above and below ground and manage to do it. So the next steps will be to focus also in endemic plant species, a congeneric species that is restricted to dolomitic soils and has a similar dispersal agent but contrasting habitat specialization. And the preliminary results suggest that the methylation levels are very different. And also from a multi-specific study, we know that epigenetic diversity, as I said, is higher than genetic diversity across populations, but only in endemic plants, both parameters are significantly correlated. What we understand is a promising result for conducting studies with endemic species. And that's it. Thank you very much. And I will be happy to answer any questions.